Assalamu alaikum. So let's get into the human genome. Now, I need to start with the telling you about this huge project, the Human Genome Project. Now, it started in the year 1990, where you have like 7 countries got together, of course, led by the United States, and they wanted to sequence the human genome. That is, they wanted to know the order of nucleotides in the human DNA in all 46 chromosomes, or let's say 24 chromosomes, including the Y and X chromosomes. And they estimated that it would take about 15 years or so, and it would cost $3 billion. Nowadays, we can sequence a human genome in about $100. So that is something about the advancement of technologies and knowledge in terms of the human genome. So the first draft was published in 2001, and 92% was completed in 2004. So this is some historical background. Of course, it was a big deal at that time, and it was announced by Bill Clinton, the president of the United States of America, and the project was led mainly by this person right here. His name is Francis Collins, and there was in the later stages there was competition by this person right here. His name is Greg Venter. He established a private company called Celera, and they were able to also to sequence the human genome, of course, using the preliminary information generated by this person and his team. So anyhow, so it was a big deal. Okay, it's like going to the moon. It's that big of a deal. So now, in addition to knowing the sequence, the order of the nucleotides in the human genome, um, there have been other major outcomes of this huge project. Um, it has enabled us to know the number of human genes, and we'll talk about it in more details in this lecture. Um, Major technologies, huge, high-tech instruments, devices, information have been developed as a result of that. And along with sequencing the human genome, scientists have been able also to sequence the genomes of other organisms, including um, advanced living beings like mice and chimpanzees and so on, or even simpler ones, different viruses, bacteria, and so on. Now, it also opened the door, it has opened the door for discussions in terms of legal and ethical issues. Can we define a person by his genome, for example? Can we give him certain incentives? Can we punish a person because his or her DNA is not good? So, there are a lot of discussions in that regard. And this is something probably in terms of ethics, that is, that I encourage you to delve into, to get into, read more about it. You can even specialize in such issues. And we need people like that. So anyhow, so let's do comparative genomics. That is, let's look and compare different genomes of different organisms. So if you look at, this is a simple table. Now, if you look at bacteria like E. coli, for example, it has one chromosome, and this chromosome is circular. It contains about 4,000 genes. That is protein coding genes. That is genes that make proteins at the end. And it has 4.6 mega base pair um, of nucleotides in that one single chromosome. Now, if you look at yeast, which we use to make bread, it has 16 linear chromosomes, about 6,500 protein coding genes. Now, let's look into fruit fly. That's the fruit fly, the Babetelfaki. So there are 14,000 genes, 8 linear chromosomes, and there are duplicates of these 8 chromosomes. Let's look at mouse. 40 chromosomes, again, doublets, and there are 20,000 protein coding genes. Now compare this to the human genome. There are 46 chromosomes, okay, doublets, and there are about, also by the way, 20,000 genes. So this number needs to be correct, needs to be corrected. Now compare this to viral genomes. Some of them are made of DNA, others are made of single-stranded RNA, and so on. There's a huge variety actually of genomes: single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA, and so on. Now, these genomes are really small, okay? Some of them even just code for one protein coding, for one protein, I'm sorry. Okay, so we talked about the mitochondria before in humans, and there is one chromosome, one mitochondrial chromosome genome, and it is circular just like bacteria, and it codes for 13 different proteins, and these are needed, like we said before, for the oxidative phosphorylation and the electron transport chain. Now again, these are higher organisms, dogs, chimpanzees, similar number of genes as human and mouse genomes. And that tells you something, you know, we all have, we sort of have the same number of genes overall. You know, we differ by very few genes. And so what does make a human human, you know? What does make mouse mouse if we have the same genes? Well, this is the mystery. This is what we need to learn more about. It has to do with the non-coding parts, regions of the genomes. And we'll talk about that throughout these lectures. So there are different ways by which we can look at the human genome. So basically, we have two genomes in our cells. One genome is the mitochondrial genome. We talked about it before. We're not going to talk about it anymore. And we have the nuclear genome. And the nuclear genome contains about 3 billion base pairs. Um, and they can be divided into two parts. We have the genes and gene-related sequences, and they make up about 30% of the human genome. And we have extragenic, that is, regions outside the genes, and they constitute about 70% of the human genome. Now, please note that these numbers are all approximate, okay? So we don't have the final definite numbers. Now, let's look at the human, the genes, the genetic part, the gene part. Now, only 1.2% can code for proteins, which is amazing. So when I first started to study, we were told that 5% of the human genome could make proteins, would code for proteins, and that was very little at that time. So in every year, this number would go down to about 4%, then 3%, then 2%, and the final number that I could find was 1.2%. And it was thought that this 1.2% is the relevant, the important portion of the human genome. Everything else was junk DNA. And it turns out that it's not really junk, it does have important value, and we'll talk about that as well. Now, if you look at the non-coding sequences that are part of the human genome, they make up mainly introns, by the way. So again, these numbers are approximate. Some of them are pseudogenes. They look like genes, but they're not really genes. They do not code for proteins. And we have other sequences like untranslated regions. So we'll talk about these untranslated regions, and we will also talk about introns later on. Now let's look into the extragenic um, sequences of the human genome. Many of them are moderate to highly repetitive. So we're talking about the same sequence, but this sequence is repeated several times in the human genome. And if we look at these repeats, we would find out that many of them are actually DNA transposons or retrotransposons. We'll talk about these two at the end of the lecture and why they are important. Now, these sequences are interspersed. What does that mean? It means that they are dispersed, repeats. They are found in different regions of the human genome. Okay, they are distributed in the human genome all over the place in different chromosomes, but these are basically the same sequences. Now, if we look at these transposons, we will find that many of them are short, some of them are long, and others are viral based or viral related sequences. We'll talk about these and their functional significance at the end of the lecture. But notice that they make up the majority of the human genome. Now, there are other sequences that can be unique. And if we look at the extragenic sequences, we would find that, we'll go back to here. So some of them are, as we said, moderate to highly repetitive sequences. Some of them are interspersed. Others, about 10% of these repeats are actually tandem repeats, which means that they come one after the other. Okay, so it's the same sequence, but it's repeated one after the other. Okay, so tandem means that they are linked, they are associated with each other, they follow each other. It's like putting your feet like this. Okay, so like that. Okay, so tandem, so one after the other. Now, if you look at these tandem repeats, we would find that some of them are mini repeats, meaning that they are small in size. Some of them are really small in size, micro repeats. We'll talk about these. And these repeats are known as satellites. Satellites, okay? For a technical reason, we're not going to talk about that. Now, some of them are really highly repetitive, tandemly repeats, randomly repetitive sequences, repeated like millions of times, and we find these in centromeres and telomeres. We'll talk about these as well. So this is the human genome, okay? And this is by looking at, this is classification of the human genome in different, in a certain way, by focusing on the sequences themselves. Near the end of the Human Genome Project, that is, at 2003, the US government started another project called the ENCODE project. And ENCODE stands for Encyclopedia of DNA Elements. Okay, and basically they want to look at DNA, and it's like taking a book and sequencing the book. That is, knowing the order of the letters. For example, you say the apple is red. Well, if you look at the letters, we're talking about THEAPPLEISRED. Well, it's meaningless, okay, if we just say the letters. The ENCODE project wanted to translate these letters into words, or to convert these letters into words, meaningful words. Okay, so they wanted to look at the human genome as such, and they found out something that is really fascinating, and that was like 10 years ago. A paper published, several papers published at that time, 10 years ago, showing that actually the percentage of the genome that is transcribed into RNA is 75%. So it's not only 2% that we were told about, the protein coding genes, the relevant genes. 75% of the human genome can be transcribed into different RNA molecules. Now, the mission now is to know what these really do. Are they meaningful or not? Are they functional or not? Are they significant or not? That's what we're trying to do. Are they just mere noise with no function? Are they disruptors? Are they functional? Do they have a purpose? That's what we want to know. And that was shocking, by the way. 75% of the entire human genome is relevant, that is, it can be transcribed. Of course, there are other regions that are not transcribed, but they are functional. They can affect the parts of the genome that are transcribed. Now, the protein coding genes constitute about 20, there are 20,000, actually, the last number is 20,000, well, 19,800 protein coding genes or such. So it's about 20,000 protein coding genes. But many of the 75% of the human genome are actually RNA molecules that can be classified into two types, long and short. And that's what we're trying to learn about. Now, there are some pseudogenes, they're transcribed, they could be noise, or they could have functional purpose, we don't know yet. So it's a really large number. About 8% of the human genome would code for transcription factors. Okay, so that's the ENCODE project. Now, in 2022, there was this study published in the journal Science where they claimed that they completed the sequence of the human genome. So what was the problem? I mean, they finished about 90%, 92% of the human genome sequence in 2003, 2004. So was it really hard finishing the rest, the 8% remaining part of the human genome? It was tough, and it was tough because of the repeats. So let's say that you have a book and the word the is repeated like hundreds, thousands of times. Okay, the, and so on. Well, and I give you the pages scattered, and I ask you to connect the pages together. How would you do that? I mean, it's all the, it's repeated thousands of times. Which one would be page one? Which one would be page two, and so on. So that was really hard. So it took them a while, 20 years, to finish the 8%, the remaining 8% of the human genome. So that was in 2022. And they were able to sequence the whole human genome except for the Y chromosome. Why? Because it is so difficult. It contains a lot of repetitive sequences, so many repeats. But let's look into here. That's this table right here. Let's look at it, and you can notice that. Let's see. So the number of genes is about 63,000. Okay, but we said 19. Well, we said 20,000. Yeah, the protein coding genes constitute about 19, almost 20,000 genes. Again, when they finish the Y chromosome, it turns out to be about close to 20,000 genes. The number of exclusive genes is about uh, 3,000, 3,600. Now, if you look at the number of transcripts, it's about 233,000 different RNA molecules, different types of RNA molecules, including messenger RNA, which codes for proteins. The protein coding RNA molecules, that is mRNA, is about 86,000. So these 20,000 genes can make 86,000 different RNA, messenger RNA molecules. How is that possible? Well, we'll talk about some examples of how cells can do that. So that was in 2022. Let's look into 2023, and it was finally done. They sequenced the human Y chromosome. Okay, so they finished. We were done with the human genome, sequencing the human genome. That was a big deal. So let's talk about the repeats now. So we'll start with the tandem repeats, and we will end with the interspersed repeats. Let's start with the satellite DNA. So satellite DNA or macrosatellite DNA, I'm not going to talk about the technical reason for why they called it satellite DNA, but the idea is that these are about 5 to 300 base pair repeats, and they are repeated like million to 10 million times. They mainly exist in telomeres and centromeres. So telo means end. Okay, so telomeres exist at the ends of chromosomes. So they contain these, repeats. We'll talk about it in a second. Centromeres are regions that exist in the middle of chromosomes, not necessarily in the center, but they are the constricted regions in the middle of chromosomes, and they divide chromosomes into Q arm and P arm. P stands for petite, small, short, the short arm. And the Q arm is the long arm, the other part of the chromosome. Now, centromeric repeats are AT rich, about 171 base pair long. They are called alpha satellites. They're unique for each chromosome. And we can, using a technique known as fluorescence in situ hybridization, we can label different regions of the chromosome. So basically here we have a chromosome and we use a probe, and the probe is labeled with a fluorescent tag or radioactivity, but basically we use fluorescent tags. And the probes would bind to chromosomes at certain regions. So you can label centromeres as shown in here, for example. You can label every single centromere. You can label, since each chromosome has its own centromeric repeat, its own unique centromeric repeat, we can use 23 or 24 different probes. Each one is labeled with a different color, and we can label each chromosome with a different color. So we can have that. We can have something that looks like this. Okay. Now we can also use FISH, fluorescent in situ hybridization, to label, to target certain genes that exist on certain chromosomes, like chromosome 5, chromosome 10, chromosome 12, etc., and to see where these regions or genes exist in what chromosomes. So this is FISH, and FISH is one of the most important techniques used to study chromosomes and chromosomal mutations, and you will study that in the genetics course. Again, since each chromosome contains specific regions, not only in centromeres but in other regions as well, we can label each chromosome with a specific color. Okay, so we can differentiate chromosomal regions from each other. Something that looks like this, very artistic. How about telomeres? Well, telomeres are made of repeats, and these repeats are TTAGGG, and these are repeated like thousands of times at the ends of chromosomes. So they exist at the end of chromosomes. Now, if we look at telomeres, we could see actually that at the end of the telomere, there is a 3' overhang of single-stranded DNA. So it's not all double-stranded. Part of it is single-stranded. And this DNA, that is telomeres, this part of the chromosome, is actually coated by a protein known as shelterin. And what shelterin protein does is it protects the ends of chromosomes from degradation, because now telomeres um, are really important in stabilizing chromosomes, in keeping them intact. If telomeres get shorter, and we'll talk about that later on, if telomeres get shorter, chromosomes become unstable and they become susceptible to degradation and mutation. Something else that is important is that there is a long non-coding RNA. So like we said, there are different types of RNA molecules that are transcribed but not translated. So they're not used for making proteins. Rather, they stay as single-stranded RNA molecules. Now, and they can be classified into two parts, either short or long. And there is a class of non-coding RNA molecules. Non-coding means that they're not used, that they do not code for proteins. There's a type of non-coding RNA molecules called long non-coding RNA. And the long non-coding RNA that is can be is there's a special long non-coding RNA that can be transcribed from the telomeres themselves, and it's known as telomeric repeat containing RNA, TERA. And there are several functions for TERA, for this RNA molecule. It maintains the integrity of chromosome termini, so this RNA prevents the degradation of telomeres and chromosomes as well. They can regulate the activity of an enzyme known as telomerase, and this enzyme keeps telomeres long. We'll talk about it in the DNA replication lecture next. And this RNA also maintains the heterochromatic state of telomeres, so telomeres are quite condensed, and the presence of this TERA bound to telomeres keeps it in a heterochromatin state that is condensed and packed. And TERA also protects DNA from deterioration or fusion with neighboring chromosomes. So it can happen, since you have this single-stranded DNA at the end of one chromosome, and you have single-stranded DNA at the other end of another chromosome. It can happen that chromosomes would fuse with each other, but this is prevented by the telomeric repeat containing RNA, long non-coding RNA. So let's look into mini and micro satellites. Now, mini satellites are also known as VNTRs, or variable number of tandem repeats. Okay, variable, okay, different numbers of tandem, because one comes after the other, repeats. So basically, it's the same sequence repeated like several times, like 4 times, for example. And we have microsatellite DNA that is also known as short tandem repeats, STRs. Now, these are simple sequences and they can be repeated several times. Again, it's the same sequence tandemly repeated, okay? Or they can be repeated 20 times and so on. So, mini satellite sequences or VNTRs are about 20 to 100 base pair long. That is, each block, each sequence is about 20 to 100 base pair long, and it can be repeated 20 to 50 different times. Okay, so you can have a VNTR here and there, and here as well, and over there. So it's this. So it can be the same VNTR, or they can be different sequences distributed throughout our genome. Now, the microsatellites are shorter. They're about 20 to 10 base pair long repeats, and they are repeated like 10 to 100 times, okay, among individuals. Okay, so you can have the same sequence, like for example, CA, CA, CA. You can have it on one chromosome repeated, I don't know how many times, like 80 times, or you can have it repeated 16 times, or you can have it repeated 14 times. Okay. Now, so we as individuals can have different repeats. So on one chromosome, the paternal chromosome, we can have a repeat STR or VNTR with a certain number, like 10, 20 times. And on the maternal chromosome, we can have the same repeat, the same sequence. On the same chromosome, we can have it repeated, let's say, 30 times. Okay. Now, we're talking about homologous chromosomes, and homologous chromosomes are basically the same chromosome, except that we get it from one from mother, one from father. So these are known as homologous chromosomes. Now, so we can have the repeats in different numbers on different chromosomes. So for example, we can have the DNA, the sequence, the STR or VNTR repeated again 20 times on one chromosome, and on the other chromosome it can be repeated 10 times. Okay, so the length, the size of this DNA fragment that contains the DNA can be different. Okay, and that is important because we can use it in forensic medicine, forensic testing, البحث الجنائي, or paternity testing, and so on. Why? Because they are excellent markers to differentiate DNA from different individuals. So if we take different individuals, 6 individuals right here, and we look at these repeats on both chromosomes, we can differentiate samples from different individuals because each one of them would have a certain repeat. So let's say individual A, individual B. Individual A is heterozygous for a certain STR or VNTR. Note again, uh, homozygous or heterozygous for this allele, the type of repeat, the type of STR, the type of VNTR, the type of gene, different alleles. So basically, if we cut this region right here, left and right, we can have two different bands. A person can be homozygous for the VNTR or the STR, specific VNTR or STR. So we can have different repeats, okay, in different individuals. And we can differentiate individuals from each other by looking at the repeats of specific STR or VNTR. So let's look into this example right here. That's a pedigree. Again, that's a pedigree. So you have male, female, and these are the grandparents. Okay, here you have a daughter, and here you have a son. They got married, And they have how many children? 7 children. Oof, 7 children. Now let's look at the distribution of VNTRs and STRs in these individuals, a specific VNTR. So we choose a VNTR that exists in, let's say, on chromosome 6. Okay. Now, there are 4 alleles. There are 4 different types of such VNTR in this family. Now, it can be different in different repeats, different number of repeats in a different family. So there are 4 alleles. Allele 1 has 1, 2, 3, 4, 5, 6 repeats of that VNTR. Allele 2, 4 repeats of that VNTR. Allele 3, 3 VNTRs. Allele 4, 1 VNTR, 1 repeat, or 1 sequence, not repeated. And let's say that we do southern blotting, okay, using a probe specific for this region right here. Of course, we cannot use a probe for the VNTR because the probe would bind to all of them, okay, and we will not be able to distinguish the different alleles from each other. So we use a probe that would bind outside. Anyhow, so regardless. Okay. So we do southern blotting, and these are the different alleles. Allele, uh, allele 1, allele 2, allele 3, allele 4. Here we have the long DNA, large DNA fragment, and here we have the small DNA fragment. So we take the DNA, we fragment it, we cut it into different fragments, and let's say that we have these fragments right here. So we have this fragment that contains this number of repeats, or that number, or that number, or that number. So here, this fragment would be really short because it contains 1 repeat. This fragment would be the longest because it contains 6 such repeats. Now, these regions right here are all identical. They're all identical. This region is identical to that. So the only difference really is the number of repeats. Okay, so we separate the DNA on a gel. We transferred the DNA from the gel to a membrane, and we add a probe. So here we have this person, this man, the grandfather of these children, has allele 1 on one chromosome and allele 2 on the other chromosome. Okay. Now, the grandmother has allele 2 and allele 3. Now, their daughter has allele 1 and allele 3. So she took allele 1 from her father and allele 3 from her mother. Okay, so we're concerned about the size of the fragment, not necessarily the intensity of the signal. Okay, forget about the intensity, the thickness of the bands. So let's look here. So we have the grandmother of the children. She has allele 2 and allele 3, just like this one right here. Okay, so yeah, you can have the same genotype, the genetic makeup being similar, identical in different individuals. So, and that's why, you know, to link, associate a sample with a person, we need to do several VNTRs, not one VNTR. So she has allele 2 and allele 3 as well. Now, the grandfather has allele 1 and allele 4. So he has this, the shortest one, and allele 1, the longest one. Now, their son, this person right here, took allele 2 from his mother and allele 4 from his father. Okay, great. Now, they got married, these two got married, and they have these 7 children, and each one of them has his or her own genetic makeup. genotype in terms of this VNTR. So note that they're all heterozygous. Okay, so this daughter has allele 3 taken from her mother and allele an allele of 4 taken from her father, and so on. Same thing with this boy right here. Now, so notice that these individuals have the same genotype. These two have the same genotype. So we need to do a southern blotting on a different region, different VNTR, different STR, to differentiate individuals from each other, or to link samples to individuals. So you can see how it turns out to be. We can do paternity testing again by looking at different VNTRs. And so this is the genetic profile, the molecular profile, not the words that I'm using. Molecular profile, genetic profile, the genetic fingerprinting, molecular fingerprinting of the mother and father by looking at different VNTRs and STRs. So this daughter right here, for example, she has this fragment, she got it from her mom, this fragment, she got it from her dad, and so on. Okay. Now this daughter right here has different profile. Okay, so she has these two that do not belong to either dad or mom, but these refer to the mother. Okay. Now, so here, this is the son of these two individuals because all of the fragments can go back, or we can link those fragments to both mother's and father's genetic profile. Now, this individual right here has totally different genetic makeup, genetic profile, fingerprint. So he is not really the offspring, not really the son of these two individuals, and so on. So um, this is it. That's how we can use VNTR and STR for. Let's look into something else, another type of variation. So all of what we've talked about so far in terms of these tandem repeats, they are variables. They are different among individuals. Something else that gives genetic variation among individuals is something called single nucleotide polymorphism. Again, polymorphism means different shapes of single nucleotides, or they're known as SNPs. So basically, these are differences among individuals, not in terms of repeats, but in terms of nucleotides, the existence, the presence of nucleotides in a certain region in a chromosome. So if you look at a certain place on chromosome, let's say 2, okay, we would find that some individuals have a T, other individuals have a C, a third group would have a G. Okay, so variations in single nucleotides in a specific place within the genome of individuals. So these SNPs occur throughout the human genome, and there are about 10 million SNPs, one in every 300 nucleotides, 300 base pairs. Now, not all of them are important. About 500,000 are really thought to be important or relevant. And we'll talk about what we mean by relevant in a couple of slides. Something else that is important is, in order to call something a SNP, a single nucleotide polymorphism, it has to exist in more than 1% of a population. Otherwise, it's called a mutation, طفرة. So really, a SNP is a type of mutation, except that it exists in a large proportion of a population. If it exists in less than 1% of any population, then we would call it a mutation in this particular population. And just like anything else, a SNP can be homozygous or heterozygous. We're talking about a specific SNP. So let's say here we have, let's say, at a certain DNA sequence, notice paternal allele, maternal allele, that is, we're looking at a specific SNPs. So on two different chromosomes, it's the same sequence, virtually the identical DNA sequence, A, C, T, G, G, A, C, T, G, G, except, and the same thing is here, they're identical, except that here we have G and G. Okay, so we say that this person is homozygous. Now another person can have G and A, let's say, and that would be heterozygous. Now a G would exist in 51% of any population, a certain population, and a T would exist in 49% of a population. Okay, but in that particular individual, this person is homozygous for G. Now another SNP is something like this, A and C. A on the paternal chromosome, C on the maternal chromosome. So we would say that this person is heterozygous for this particular SNP and homozygous for this particular SNP. Now A exists in 90% of the population and C would exist in 10% of any population. So that's the major allele, that's the minor allele. Now if we look at, again, chromosome 2, okay, maternal individual 1, individual 2, 3, 4, 5, 6, etc. We will find that individual 1 has TA or CJ. So this person is heterozygous. Now individual 2, CG, this person is homozygous for this particular SNP. This person is homozygous, but this person has a TA and TA, okay? And so on. This person is heterozygous, heterozygous. Now note that here we have, let's say, the paternal chromosome, and here we have the maternal chromosome of this person right here, and so on. So these are SNPs, variations, genetic variations. Now, what is the significance of SNPs? Well, it depends on where the SNP exists. If we look at the human DNA, we would find that we can sort of divide it into different regions as well. So we can say here we have the coding region. This is the region that makes that codes for a protein. This is a region that regulates the expression of the coding region. So this part is not necessarily it does not code for a protein, but it regulates the expression, the activity of the gene, the coding region, that is. Now other regions may not have any functional significance. Okay, so SNPs in regulatory sequences or coding regions can be significant, and they can cause a phenotype. They can cause a phenotype. So for example, if we have, so we call them causative SNPs. So if a SNP, a variation, exists in the coding region, this can result in the existence of a different amino acid within the protein that is made from this gene. That's a different amino acid that can affect the protein function. Okay, so we call this again a causative SNP because it causes a phenotype. The enzyme may be less efficient, it may be mutated. Well, you know, it can make a person having green eye color, a blue eye color, and so on. Now, if the SNP exists in the regulatory sequence, it can affect the expression of the protein from the coding region. And that would cause, again, a SNP. So the enzyme, for example, that is made from this gene, from this coding region, may not be expressed at a high level enough to make the person normal. And that is why, if you look at any population, you can see that there is some variation. Some individual may require one pill of Panadol. Another person would require two pills. One would not be enough. Now, they all have the same enzyme, they all have the same gene, but maybe in one person the gene is less efficient, it's not expressed well. The enzyme does not work as efficient as if there is this other amino acid, and so on. So SNPs may cause genetic variations among individuals in populations. Okay, so that's why, oh, that's causative SNPs. So causative SNPs occur in coding regions or regulatory regions. Now, and you can read these, by the way. Now, some SNPs may not be relevant whatsoever. Majority of SNPs may not be relevant, but some of them are known as linked SNPs. They are not causative. So by having, for example, an A or a T or a G or a C in a regulatory sequence or a coding region may cause a phenotype. But these linked SNPs do not necessarily cause a phenotype. They do not make a person need two pills instead of one, or have an eye color or have an eye color of a certain color. Okay. It's just that they are linked. We say that people who have A and in this part would need one pill of Panadol. Okay. Well, what if we change A into something else? Would that, in that particular person, would that make a person need two pills instead of one? No. It's just that people who have A need one pill. That's it. Just linkage. Okay. So, so the example that I always use is that let's say that there are two people, two independent people. They're always together. They always walk around together. Always. So if we see one person, we would immediately say, ah, the other person will just come into the room right now. Why? Because he is linked to that first person. They are always together. Having one person means that we're going to see the other person. It does not mean that they cause each other, or that they are identical to each other, or that one of them, if one of them exists, the other one exists as well. No, it does not mean that. It just means that they are linked, they are associated together, they are always together, or there's a high probability that we're going to see the other person. So, for example, there are certain diseases, we don't know what the causes, the genetic causes of these diseases, but we know that patients who have this particular disease have a G in this part of the genome. It's just G is associated. It does not mean that G causes that disease. It just means that by having a G, there's a good probability that this individual may have the disease. It's just that that G is linked to that disease for some particular reason. It could be that the gene that causes that disease is close to the G. So when they are inherited, when this gene is inherited, it's also inherited with the G. If the gene that causes that disease is inherited by an individual, the G is also inherited with that gene that causes the disease. But we don't know what gene causing that disease. It just It's just a marker. G would be a marker for having that, for the increased, for the higher probability that the disease will be will affect that individual, or will exist in that individual. So linked SNPs are outside of genes. They have no effect on protein function or protein production. If we change them, nothing is going to happen. It's just that they are linked. That's it. Let's look into interspersed repeats now. So the majority of interspersed repeats are transposons. Transposons are also known as jumping genes because they change places. They keep on jumping from one place to another in any genome. And there are two classes of these jumping genes or transposons. We have DNA transposons and we have RNA transposons. RNA transposons are also known as retrotransposons. So when we say retroviruses, retroviruses are viruses whose genome is actually RNA, not DNA. So these are known as retroviruses, just like the HIV virus that causes the AIDS. Now, the majority of the human genome is made of retrotransposons. RNA got integrated into our genome millions of years ago for some unknown reason. DNA transposons make up 3% of the human genome. Retrotransposons can be divided into different types, two different types, either long interspersed elements or short interspersed elements, or lines and signs. The majority of retrotransposons are lines. An example of a short interspersed element is a sequence known as ALU sequence. It's about 300 base pair long repeated sequence, and this ALU sequence exists all over the human genome. It can be integrated into protein coding genes as well. Now, we have retrovirus-like elements. They make about 8%, 10% of the human genome. So I said that now these transposons are jumping genes. They keep on jumping from one place to another, but it's not really that I'm talking to you right now and my transposons are jumping all over the place. Actually, the majority of transposons have lost the ability to jump, to change places. So we don't have that happening in our genome. But it does happen in other organisms, I think like pigs, for example. So anyhow, so this is the story of transposons. Okay, now why do they exist in our DNA? We don't know. What do they do exactly? We know something, but not the whole story. So these retrotransposons, how do they move? How do they integrate? How do they change places? Well, it turns out that some retrotransposons can be transcribed, producing an RNA molecule. And this RNA molecule is converted by an enzyme known as reverse transcriptase. Reverse transcriptase. This is an enzyme that is coded by some retrotransposons. So some retrotransposons can code for reverse transcriptase. And this reverse transcriptase changes RNA to DNA, double-stranded DNA. So transcription is basically making RNA out of DNA. Reverse transcription is making DNA out of RNA. Now, once the RNA is reverse transcribed into DNA, this DNA can be integrated into a different portion of the human genome using an enzyme known as integrase. And this integrase can also be coded by certain retrotransposons. So the majority of retrotransposons do not have reverse transcriptase, do not have the integrase gene. Some can make integrase and reverse transcriptase. And this allows for a transcribed retrotransposon to for the RNA of that reverse the RNA of the retrotransposon to be transcribed and reverse transcribed into DNA, and the DNA to be integrated into the human genome at a different place. We still don't know the full story of these enzymes and what they do and how they work, but scientists are working on it. So this is really fascinating. So 99% or more of the transposons cannot move. They're just there, sitting there. Some of them can be transcribed into can be transcribed and translated, producing proteins. Some of these proteins or examples of these proteins are reverse transcriptase and integrase that can help in moving the transposons from one place to another. Now, what's the outcome? What's the effect? What's the significance of having these transposons changing places? Well, it depends on where this movement takes place, where this integration takes place. So the transposon can integrate within a gene, making the protein product defective. It's not functional anymore. It can integrate into the regulatory sequence, like the promoter sequence, the enhancer region, and it can affect the transcription, the activity of that gene. We'll talk about that in the transcription lecture. Or it can integrate somewhere else, intergenic, outside of genes, outside of regulatory sequences, with no effect whatsoever. Okay, so it depends on where the transposon integrates into the genome. And it turns out that there are certain diseases like hemophilia, like SCID, severe combined immunodeficiency, porphyria, different types of diseases. Duchenne muscular dystrophy, for example, they can be caused by having transposons integrate into these the genes that if these get defective, different diseases can be caused. Still, this is under extensive research, and we will know more about it in the next years or so. Allah